Hello, everyone. Uh, I'm Rafi. I'm a PhD student from the School of Biomedical Engineering at the University of British Columbia. I'm from the DEBOR lab. And today I'm going to present our work detecting and avoiding homology-based data leakage and genome-trained sequence models. And this is a collaboration with Brett Kyoto from Yachi Lab. They're also at our department. So by genome-trained sequence models, I'm referring to models like CromBPNet, Informer, Borzoi. And these models are trained to predict gene expression or epigenetic states from DNA sequences alone. And these models take in 2000 base pair to half mega base pair of DNA. And they're predicting molecular phenotypes across different cell types or tissues. So when we train these models, our goal is that the model would learn the molecular underpinnings or the regulatory logic that dictates how genes are expressed across different tissues and the epigenetic states. But when we test these models, what we don't account for is that within the same species, we can have homologous sequences across chromosomes. So here, I'm showing you an example of a 1000 base pair region from chromosome 16 and chromosome 9. And these are highly homologous, like Smith-Melamed score would be almost like, I think, 900 for this pair. And you can see that they're sharing the accessibility profile. What you don't want to do is train on one of the sequence and test on the other, because if you had a overfit model that is memorizing, in this case, you'd be making a perfect prediction, which couldn't be based on the underlying regulatory logic. So just to make my point clearer, I'll show you a simulation here. So I just created a set of sequences, and these are just random sequences, 200 base pair long. And in one set of sequences, I inserted 1 to 3 transcription binding sites, I think 10 base pair, 20 base pair long. And I just randomly put them throughout the sequences. And the other set, they do not contain any TFPS in them. Then I created homologous sequences from these base sequences pairs. And the sequences that have TFPS, I level them as 1, and the sequences don't have any TFPS, I level them as 0. Now you can guess that the task I'm going to do here is that I go and try to train a predictive model, and the task for the model is, can you identify if the sequence contains a transcription binding site? So the first attempt I do, I train a single layer CNN model, and I drew pretty good because it's a very small model. I get 0.7 AUC. So at this point, any machine learning engineer, they would actually go and try to tune their model further, try something different. So I try increasing the capacity of my model, and I seem to do pretty good. And then I say, what the heck, let's try something bigger. And I train a even larger model, and I'm almost approaching 100% accuracy. Now, how am I evaluating the performances here? I have this data set. I just split it randomly, train and test, and I'm evaluating the performance on the test set. Now, if I created a homology-aware test set in this setting, this is how the performances look like. And what this tells me now is that I have not yet solved the problem. The logic I wanted to learn was that, do I have a TFPS in the sequence, which I have not yet learned. So the next step is that, okay, I have to improve my modeling, or I have to go and get more data. So this connects with what John Jumper said in his keynote, and not the one about where you all have 256 GPUs, but the point where he was talking about that in DeepMind, they had a very good test data where they were testing AlphaFold. And whenever they were trying out different things, and they were trying to maximize the performance of the model on the test set. So it's very important to have a test set you can trust. And when you have a test data you can trust, you can go on and try out all these different things. And as you improve your performance in the test set, you know that, okay, you're solving the problem. Now, you'd be right to not trust the simulation alone, and you may want to see what happens when we go and apply trained models on actual genomic data sets. So I'm going to show you what happens in a very controlled setting. And so the data sets I'm going to work on here is a lentium pair data, where is from Nada Vahitov's lab. They have annotated 226,000 short sequences in K562 cell line, and these sequences were promoters and putative enhancers based on their accessibility profiles. And this is a very controlled setting, so the activity derives from the sequence content alone of these short 200 base pair sequences. So anything I observe here would be worse if I did it in genomic context, where the context is changing and you have more homology. And another important point is that these are 200 base pair sequence, so I could feasibly calculate the exact Smith-Roman element score in a pairwise manner. So as a metric as a measure of homology, we use Smith-Roman alignment score, which can be thought of as the gold standard for calculating local alignment region between any two sequences. And as a metric for leakage between two sets of sequences, we use maximum Smith-Roman alignment score. So this is for sequences from set 1 and set 2. You'd calculate all the Smith-Roman alignment score, and the maximum score you get, that's the degree of leakage for each sequence across the other set, against the other set. And we also design a model like that is, there is no idea about system logic, but what that model does when it is asked to make any prediction, it looks at the most similar sequences in the sequence in the training set, and it will take an average of the expression and just make the prediction. So it is kind of like, giving you the expected predictive ability of an overfit model that has no idea about the underlying logic. Now I go and train different neural networks and apply overfit NN on this lentium pair data. And I split the sequences chromosomally. I train with some chromosomes. I'm testing with the other chromosomes. But I've already calculated the local alignment scores between the train and test set. On the x-axis, you're seeing the degree of leakage using which I'm binning the test data. And on the y-axis, you're seeing the performance of the models. So what you see here is that on the distant homology range, whereas there's no similarity, something like overfit NN wouldn't perform, but the neural networks do perform. So yeah, the models are learning something that are predictive from the sequence content. And as you go further closer into the higher homology ranges, you see that overfit NN performance is increasing. And at the highest homology range, sequence content alone can be used to predict expression, showing that these are not that hard to predict sequences. But we see another interesting thing is that if you ignore this part, because this is where the sequences are more distant, but you see this trend where you might have expected the performance to go down as you move along the homology distance bins, but you see this uncanny valley. And this would become relevant later on, in a few slides later. But one could claim that maybe in this middle region, the models are trying to use memorization to predict, but the sequences are different enough that the regulatory activity is different. So that's why the models fail in these regions compared to even more distant bins. And we see another interesting phenomenon that on the test set, on the highest homologous regions, sequences, models fit very fast during training. So x-axis is epochs as we are training, and y-axis I'm saying that how these different ranges of test sequences, how models fit to them. And we see that the highest homologous ranges, models fit very fast, even faster than the overall training data. Now, what happens to models like ChromeBPNet or Informer or Borzoi? And because we're very much interested in knowing that the features they're learning, are they generalizable, or are they learning something that are not, that it doesn't have to do with causal security logic, something Maria Chikina highlighted in her talk, I think 2 days ago. But quantifying homology for these models and longer sequences is a harder task, because we want to be calculating the something like Smith alignment scores, but the computational burden would increase quadratically with sequence length and the number of sequences. So to this end, we developed a tool, HashFrag, which can does these things for you. And it basically uses BLAST on the backend. We tried using other methods on the backend, but BLAST came out victorious at the end. And so using BLAST, you can, if you have a set of sequences, you can create a database with those sequences and query the sequences against the database. And it will give you all the candidate similar sequences, and along with the Smith alignment scores, and you can use that.
No, we lost everything.
Okay.
No, honestly.
No worries.
Let's start again.
I didn't step on anything by the way.
But
Yes.
let's try again. It had to happen eventually. Go ahead.
This is fine. So yeah, so you can detect the homologous sequences, and using the scores, you can filter for high homologous sequences in your existing train test splits. You can, another exciting option, and I think which is most impactful, is that you can stratify the test sequences by homology and see if there is any memorization built into the network if you do a lot of subsequent analysis. And another another application is that you can go on and create homology-aware train test splits. So in this figure, I'm just going to brush through some slides here, how HashFrag splits do not have the train test leakage in terms of homology scores. And here I'm showing how, so each row here are different chromosomal splits, and each column is a different model architecture. You can see that using HashFrag, how we were able to remove homologous sequences from the test set at different thresholds, and how it leads to removing overestimation from the model. Now, previously I mentioned that it was not feasible for us to do the Smith-Riemann calculation between the train and test set for something like CROM-BPNet, because I calculated it almost would have taken something in the range of 50 CPU years. But with HashFrag, it was something that was feasible to run. And we calculated the Smith-Riemann element score between the train and test set for one of the folds of CROM-BPNet. And we were trying to see, okay, what happens to the performance of the model in different homology distance bins. And we see something very interesting, something that also showed up in the MPR model performance, that the models are doing good on the lower homology, higher homology range, and the lower homology range even. But in between, you see this uncanny valley showing there might be a competition between memorization and generalization in these regions, where the sequences are similar to some training set sequences, but it's distant enough that the activity is different. And here, the predictions I'm working on is the bit count head from CROM-BPNet. Now, I'm just going to talk about some of the problems that we will have with applications of these models. And one of the big promises of these models is that we want to make variant effect prediction with them, specifically for the GWAS variants. Now we know that homology can appear across chromosomes. Oh, I will take probably 2 minutes. Yeah, 2 minutes more. Yeah. So homologous sequences can appear across chromosomes, and we can have variants that can lead to phenotypic changes. Now think of some of these variants, they can also appear across chromosomes. And if you think of the alternate allele, possibly they could be appearing across chromosomes as well. So I went to open target dataset. I filtered for high confidence SNPs based on the PIP score. And what I did, I took 20 base pair regions here surrounding the alternate allele, and I tried and searched for those 20 base pair regions in other chromosomes in the genome. And apparently 60% of them appear somewhere else. If you go and look for 40 base pair regions surrounding the alternate allele, 6% appear somewhere else. So, and here I'm just showing you how many times they appear, and some of them seems to be appearing more than once and quite a lot. And it's not like they are only appearing across chromosome pairs, but they seems to be appearing across all chromosomes, where maybe the source is chromosome 1, but you do see them across all other chromosomes. So the problem is, if a model has seen these alternate alleles during training, even if there is memorization in the model could be predicting the variant effect correctly just based on memorization alone. So this leads to a problem in evaluating these models in variant effect predictions when you have homology-based data leakage. And this is one of the last applications that we had for hashfrag tool, is that what if we create homology-aware trained test splits, what would happen? So what we did is that, by the way, one problem is like of evaluating hashfrag splits versus chromosomal splits is that when you test them on chromosomal data, because this data comes from the genomes, is that any chromosomal split models would have a benefit by default. So you just may have an overestimation. So we evaluated them on synthetic data from Guasa et al., and specifically sequences which look substantially different from genomic DNA. And on that test set, we saw when we trained with hashfrag splits, they outperform chromosomal split trained models. And lastly, I want to conclude by highlighting one key point, is that homology between training and test sequences will compromise the reliability of your model evaluation. So we should try and solve that as a field. And in future, I plan to do the analysis of memorization on longer context models, which is a much harder problem. Hopefully I'll be able to do that. And another exciting thing would be if we can provide pre-competitive scores for the whole genome in a genome versus genome manner by creating bins and comparing it against every other bin of the genome, then it would be very easy to do any sort of analysis for any models, just so you don't have to run the same thing again and again. And yeah, that's the end of my talk. I want to acknowledge my amazing lab members.
Thank you, Rafi. Questions?
Thank you very much. Really nice talk. When you looked at the ChromeBPNet results, I was very surprised to see that actually on the very low similarity on the right-hand side, you have way better results than on the very high similarity.
Yeah.
Oh, sorry, it's going back a lot. Here. I mean, if you look at the end side here and compare to the second bin on the left.
Yeah, it's even like, higher than the most homologous bins. Yeah.
Yeah, and it's the same almost as in the middle. So do you have any hypothesis why this is going on?
I'm not so sure, but I can give you some examples. Is that in this bin, you have a small number of sequences because there are not like thousands of sequences that fall in that range. So that could be one of the reasons that why you have like, this discontinuity. So yeah.
Thanks, Rafi.
Yeah.
Is it feasible to run hashfrag on 500 KB or a megabase regions?
So for a 500 kilobase per region, it doesn't make sense for two sequences to have a shared ancestry. So it wouldn't be like directly, or these two sequences are homologous. But as I mentioned, that so we were planning to actually pre-compute the score. So the way we would do it is beam the genome into, let's say, 1,000 base pair tiles, and for 500 KB region, you will have a tiling score of what is the degree of leakage for this specific region. And what you could do, you could remove specific tiles during model evaluation when you report your Pearson R on the test set.
Yeah, I think there's a lot of tricks you can use from sequence analysis to speed this up. Yeah, I think there's a lot of work. If you look at the literature, and if you look at maybe what is reported in HitSeq, you talk to these colleagues. There's not a problem to do that, to pre-compute this for us for all the genomes, and it's, I think, something we'll have to do. So thank you very much for this interesting insight, Rafi.